Evolution of seed. Fertilized and ripened ovule is known as seed. Seed can be survived under unfavorable condition. It is responsible for the preservation of species and it made the plant's dominant vegetation on land. If we look into those plants which do not bear seeds, like lower vascular plants, those plants become reduced in number with the passage of time due to the lack of seeds, because their sporophyte and gametophyte does not preserve itself under the unfavorable condition. Seed plants, for example spermatophytes, is now the dominant vegetation on land, and this is due to the seed. How the seed evolved in plant when none was present. Steps in the evolution of seeds. The first step in the evolution of seed is the development of heterospory. What is heterospory? The production of two different types of spores is known as heterospory. In heterosporous plant, there are two types of spores: small spores and large spores. Small spores are known as microspores, and these are produced in the microsporangium, while a relatively large spore are known as megaspore, and they are produced in the megasporangium. If we compare the seeded plants with the non-seeded plants, the non-seeded plants are homosporous. It means that they have a single type of spores, and the spores was not differentiated into microspore or megaspore. So the first step in the evolution of seed is the production of two different types of spore, which is called heterospory, or development of heterospory. After this step, the megaspore was reduced. If you look into the megasporangium contains many megaspores. During the passage of time, these megaspores are reduced in number and a single megaspore is retained inside the megasporangium. The third step is endospory. What is endospory? Endospory is the retention of megaspore inside the megasporangium. If we look into the homosporous plant, in those plants the sporangium bursts and all the spores are released from the sporangium. While in heterosporous plant, only the microsporangium bursts and microspores are released from the microsporangium, while the megasporangium remains intact and the number of megaspores are reduced to 1, and this single megaspore is also retained inside the megasporangium. It means the megaspore did not shed from the megasporangium. This is called endospory, or retention of megaspore inside the megasporangium. The final step in the evolution of seed is the formation of integuments around the megasporangium and evolution of ovule. If you look into the megasporangium, with the passage of time, this megasporangium is covered by leaf-like structures. These leaf-like structure then fused with the microsporangium, and these leaf-like structure become hard to protect the megasporangium. These leaf-like structure are known as integuments. So the integumented megasporangium is known as ovule. Evolution of pollen tube. Pollen tube evolved parallel with the evolution of a seed. This is the pollen grain. When the pollen grain flow towards the ovule or integumented megasporangium, it land on an opening in the ovule. This opening is known as micropyle. If you look into the pollen grain, the nucleus of the pollen grain cannot reach to the ovum directly. Rather, the pollen grain produce a tube-like structure to transport its nucleus toward the ovum. This tube-like structure is known as pollen tube. The pollen tube transport the sperm nucleus from the pollen grain to the ovum, where it fertilizes the ovum, and this fertilized ovule is now known as seed. As we defined earlier, that seed is the ripened and fertilized ovule. This is the embryo, and this is the nutritive tissue inside the seed, which is called endosperm. Seeded plants are the most successful land plants because it did not need water for fertilization. If we look into the seeded plant, the water factor is eliminated by the evolution of the pollen tube. That is why the seeded plants are present in all type of habitat on the earth.